Our next speaker is Sarosh Nagar, and he will be presenting Determining the Size of the Useful Field of View Based on Target Object Attributes.
So I'm going to start with a bit of a problem. This is a mammogram in which a radiologist looked in green. The breast cancer microcalcification in this image is in red. When the radiologist looked at this image, they missed the cancer. This type of misdiagnosis occurs about 20% of the time and contributes to about 40,000 deaths every single year. It also triggers thousands of car accidents, and all of them stem from the same purpose, which is that you do not process some of the things you're seeing. But if we could calculate how much of an image you actually processed and could understand in your brain, we could use that as a metric to say you might need to look at this region of an image in order to maximize your coverage and avoid missing future targets. And so my project specifically seeks to analyze how that useful field of view, the area you see and processes, changes depending on the target object you're looking for in terms of its size. So a couple visual basics. When you stare at any point, so you might be staring at the slide right now, you're fixating on it. When you decide you're inevitably bored of this presentation, you'll move your eyes somewhere else and make a saccade, a quick eye movement, to another fixation, which is another point you'll stare at. We measure how long all of this is in the basic visual unit of the scan path, which is composed of all things and is how you see everything in the world. Its length, my bad, is measured in terms of visual angle degrees. A good way to think about this is if you were to stick your hand out in front of you, but don't hit the person in front of you. Just stick your hand out in front of you and go from one end of your thumb to the other end of your thumb. That's about 1 visual angle degree. So to illustrate the useful field of view from that, we have a little bit of a demonstration here. I want everyone to stare at this red dot. I'm going to pull up an image to the right of the red dot, but you need to keep staring at the red dot. And if you can perfectly see the image I pull up to the right, clap once. What if I move it here? That is your useful field of view. It's the area you clearly see and process because it correlates to the fovea, the region of your eye with the highest number of retinal scanners. For outside that useful field, things tend to become a lot more grainy and blurry. If you stared into the red dot at the center of this image, you'll notice your perception goes down pretty badly because you don't have as many retinal scanners in your visual periphery or everywhere outside the useful field. So we sought to measure, well, how does this clear area in the center, this useful field, change depending on what you're looking at? So we ran a 4-stage experiment with 9 subjects. And there's no better way for you to understand what the experiment is than for you all to do it yourself up here. So on stage 1, we're trying to measure, well, how does a big, easy-to-see object change your size of your useful field? So behind me on this image is going to be a bunch of large black letter L's and T's and vowels. And I want you to find the one letter T. And when you do that, clap your hand. The letter T is right here. And in this case, we contrast the ease to find those large objects with what we'll contrast in stage 2, which is looking for a smaller, blue, or not easily as seen object. So in this case, we had people look for A's and E's, these small letters that are much harder to find and clustered together. So in the next image, there will be an A, and when you find it, clap. The A is also fairly easy to see, but because of how clustered it is, it makes it more difficult for you to find it along the way. In stage 3, we then sought to measure, what if you average and you're looking for multiple targets? How does that change your useful field size? So here, in particular, there will be a T and a letter E. If you find the T, clap, and if you find the E, stomp. In this case, testing multiple object presence is trying to see how that would affect the useful field size. And in stage 4, we don't really have demonstrations because we had the same thing as stage 3, except we added value to a target. So we told people basically, this E is worth 2 points, this T is worth 1 point, and you need 200 points to finish this. Whereas in every other experiment, we had you do 200 trials to finish the experiment. Along the way, we used an eye tracker to help track everyone's eye movements. And we calculated the size of the useful field through what we call the last saccade, or the last eye movement measure. So when you're moving and looking, and you find a target, the last eye movement you make to the target is the radius of your useful field, at least as we specify. Because in order for you to be able to realize that that T was your target, you need to process it. So it at least needs to be inside your useful field, which means measuring from this point as a radius is a good conservative measure of the size of your useful field. To help verify our methodology, we plotted this frequency graph. And basically, along each of these plots as well, this is the last eye movement, the second to last eye movement a person made, and every other one. And we said, how long is this eye movement? And then how often was it this long? So in this case, the last eye movement was a length to about 23% of the time. This correlation with the last eye movement being longest the most is a good way to say that it's consistent. So it's a good measure of how your useful field is. To help verify this, we had a computer simulation help perform part of the experiment. We wrote a MATLAB code to basically have a predetermined useful field size of 1 degree, 10 degrees, and then we took the human's data and literally had the computer try to search as if it were a human with their data. With this process, the computer would eventually find its target, and we plotted the very same graphs again. When we plotted these graphs specifically, you'll see that in general, when the useful field is about 1 degree, or if you could only see and process things the size of your thumb, the graphs are all kind of clustered together, which kind of makes sense because that would be a very silly way to view the world. In stage 10, the model absolutely looks nothing like it. All the other I measures tend to be a lot longer and more specified versus our specific instance. But if we plugged in the human data, the correlations and trends seem very similar. Sure, in general, the values are different, but we find that the useful field of view in your last eye movement tends to be the same throughout. So with this method being verified, we can say that the human data we tested helps support some of our methodology. So in terms of actual results, we find that the average useful field of view in everything you look for is about 5.2 degrees. That's pretty standard with previous literature on this topic, which might support some of the other conclusions we come to. In particular, when we split it up by target object, we find some really interesting things going on. First, when you're looking for your big, black, easy to see, or distinct letter T, your useful field is the largest, likely because you kind of need to zoom out a little bit to process the entire image. If we contrast that where you're looking for the A's and the E's in this stage, you can see that because they're a lot smaller and they're blue and they're clustered together with other vowels, it makes your life more difficult as a result of it. So your useful field shrinks down a bit to correlate with a smaller object. If you're looking for the T's and the A's, your brain is basically playing the Goldilocks game and deciding that between the A's, the E's, and the T, you kind of need to average your useful field size to try to look for as many of the multiple targets as you can. But in stage 4 is where we get some really interesting things, because as soon as we added some point values to this, your useful field size went down quite dramatically. So we ran a series of pairwise t-tests against stages 3 and 4, because the two stages here are the same except for that point valuation, and we find the difference of adding psychological value to be statistically significant. What that means in this case is we have evidence that when you add psychological value to the target, your literal anatomy at which you see things changes. That's pretty unprecedented, given that most evidence we find about this says that maybe the target you're looking for or how quickly you look around will change when you look mad or angry or you're looking for something really hard. But here we can see that that useful area you're looking for is adjusting to what is more valuable for your eyesight. The downside of this, and sort of the contrast with this, is if you're looking and you're adjusting for high-value targets mainly, you run the risk that low-value targets that you aren't adjusting your useful field for, you might end up missing them on the way. And this is something important to discuss. So in terms of future applications that we find in some of this work in general, they probably have two broad applications. The first is probably best explained by the driving diagram on the left, which is we can calculate, well, how much of an area do you actually process? When you can calculate, well, that you looked at this region of an image and you could only process about 30% of it, we could educate and train people in the future to look at specific parts of an image, or in the case of a mammogram, look at a specific region of the breast in order to say, you need to look at this area to maximize the component of the useful field. The second thing it also highlights is that if you're trying to look for highly valuable targets, there's some incentive to having secondary reviewers, people who don't know the value of a target but just know what the target are, look at your targets and values again in order to make sure generally that you aren't missing low-value targets while you're searching for high-value ones, such as you wouldn't necessarily, if you were looking at a lung CT scan, for example, don't miss a secondary infection while you're looking for a late-stage tumor. In conclusion, we find that the useful field of view tends to increase as the size of an object gets larger and becomes more distinct, and goes down in the opposite way. That when we average multiple targets, your useful field of view tends to average itself accordingly. And that when you add high value to a target, your useful field of view in general looks for these high value targets. In the future, I'd say work could most consistently go towards seeing how clustering would affect the useful field. So like if I was looking for Braden in this case, in this massive cluster of people, it's harder for me to identify him because he's surrounded by so many non-targets with a target. So we have to look for him extra hard. The second thing is seeing how other emotions, so like stress or sadness or anger, would change your useful field. Because if value can have an impact on how your useful field changes, other areas might also have an effect. I'd like to thank a couple people with whom this work would be invaluable. So my mentor, Professor Wolfe, and Dr. Wu for making this incredibly amazing, and honestly, I've learned so much. Dr. Silman, Jenny, and Deborah for helping read and see my presentations. Caitlin and Dave for being wonderful people to work in lab with. I'm really sorry I annoy you every day on the morning to lab, but regardless, there. And Emin for being a wonderful counselor, and everyone else. And RSI, CE, MIT, and the sponsors.